Imagine a world where the code of life is no longer a secret, but an open book, one whose pages we can finally turn, study, and even rewrite. For centuries, the mysteries of our biology have been locked away, hidden deep within the spirals of DNA, waiting for us to discover their secrets. Today, that book is in our hands. We can read the text of life, and for the first time, we hold the tools to edit its chapters, to correct its errors, and to imagine new possibilities for ourselves and future generations. We now have the power to duplicate living beings, to create life that mirrors itself, and to push the boundaries of what was once thought possible. With gene editing, we can alter genetic instructions. Abilities once reserved for science fiction, now becoming reality in our laboratories and clinics. Humanity stands at a crossroads, able to direct its own evolution for the first time in history. The choices we make today will shape not only our own lives, but the destiny of generations yet to come. This journey into our biology is one of breathtaking promise and profound peril. We are explorers on the edge of a new frontier, filled with hope, but also with uncertainty and ethical dilemmas. We used to look to the stars for answers, searching the cosmos for meaning and our place in the universe. Now, we look within ourselves and ask, what does it mean to be human, when we can change the very fabric of who we are? The tools we create today, powerful, precise, and full of potential, will shape the future of our species in ways we are only beginning to imagine. We are learning the language of DNA, a language billions of years in the making, written in the code of every living thing on Earth. With this knowledge comes the power to cure ancient diseases, to heal wounds that have plagued humanity for generations, and to offer hope where there was once only despair. But with this power also comes the ability to alter our very essence, to redefine what it means to be human, and to face questions we have never had to answer before. The conversation about cloning and genetic engineering belongs to all of us, not just scientists. It is a dialogue that must include every voice, every perspective, and every hope and fear. The choices we make now will echo through generations. Will we conquer disease, extend life, and unlock new potential? Or will we risk losing a part of our humanity in the quest for perfection, forgetting the value of our imperfections and the lessons they teach us? The future is not something to wait for, it's something we must choose, together, with wisdom, compassion, and courage. The story is just beginning. The next chapter is unwritten, and its possibilities are as vast as our imagination. And we are its authors, each of us holding the pen, ready to shape the future of life itself. In 1996, the world changed with the birth of Dolly the Sheep, the first mammal cloned from an adult cell. Dolly's arrival was more than just a scientific milestone. It was a moment that captured the imagination of people everywhere, sparking debates and dreams about the possibilities of cloning. Scientists achieved this by taking a cell from an adult sheep, carefully removing its nucleus, and fusing it with an egg cell that had its own nucleus removed. This delicate process created a genetic copy, a true duplicate of the original sheep. Dolly proved that DNA from a specialized, fully developed cell could be reset, reprogrammed to create entirely new life. This discovery shattered the old belief that only embryonic cells could generate new organisms. This process, known as somatic cell nuclear transfer, became the foundation of reproductive cloning. It's a technique that has since been refined and repeated in labs around the world. In essence, the nucleus from a body cell is transferred into an empty egg, which is then stimulated, often with a tiny electric shock, to begin dividing and developing into an embryo, just as if it had been fertilized naturally. Since Dolly, scientists have cloned a growing list of animals, from mice and cows to pigs and even monkeys. Each success has shown our increasing mastery over the code of life and has opened new doors for research in medicine. These experiments aren't just about creating copies, they help us study disease, understand aging, and even offer hope for preserving endangered species by cloning animals that are at risk of extinction. Today, the science of cloning is no longer about asking if it can be done, but how and when it will be used. The focus has shifted from possibility to practicality, and from theory to application. But Dolly's birth also raised a profound and unsettling question. If we can clone animals, could we one day clone humans? The idea moved from science fiction to a real, if controversial, possibility. The technique is remarkably similar across species, making the cloning of humans not just a theoretical concept, but a genuine scientific possibility that some believe could happen within our lifetimes. But with this breakthrough came a host of ethical challenges. Scientists, ethicists, and the public began to debate the consequences and responsibilities that come with such power. Are we truly ready for a world where a living person could be a perfect genetic copy of another? What would it mean for identity, for family, for society as a whole? Dolly's legacy is not just scientific, it's a mirror reflecting our deepest hopes and fears about what it means to be human, and about the future we are shaping with our discoveries. The question is no longer about technology alone, but about humanity, about our values, our choices, and the kind of world we want to create. What will we choose to do with this extraordinary power? Will we use it to heal, to help, or will we cross lines we cannot uncross? The answer to these questions will define not just the future of science, but the future of humanity itself. Human cloning faces near-universal resistance, not just from scientists and lawmakers, but from people across cultures and backgrounds. The idea of creating a genetic duplicate of a person raises profound questions about identity, individuality, and what it means to be human. That's why cloning is banned by laws and international agreements in almost every country. The main reason is safety, protecting both individuals and society from unknown risks. Cloning is inefficient and risky, with many failures and health problems in animals. Most cloned embryos never survive, and those that do often suffer from severe genetic defects, shortened lifespans, or painful medical conditions. Attempting this with humans would be unethical experimentation, exposing people to suffering and loss without their consent. Beyond physical risks, there are deep psychological concerns. The emotional impact on a cloned individual, and on families, could be profound and unpredictable. What would it mean to grow up as someone's genetic copy, living in their shadow? Would you ever feel truly unique, or always compared to the original? Cloning could turn people into commodities, sources for organs, or tools for immortality. The temptation to use clones for spare parts or to fulfill someone's wishes is a slippery slope. This threatens human dignity and risks creating a biological underclass, where some lives are valued less than others. The ban on human cloning is an act of humility, recognizing limits to our wisdom and the dangers of unchecked ambition. We can read the book of life, but we barely understand its meaning. Each discovery brings new questions, not just answers. To clone a human would be an act of hubris, assuming control we do not possess. The consequences could be far-reaching and irreversible. As our power grows, so must our wisdom. We must ask, not just what we can do, but what we should do. Some boundaries exist for a reason. Respecting them is part of what makes us truly human. Cloning takes two paths, reproductive and therapeutic. Reproductive cloning aims to create a full organism, a genetic duplicate. This is widely banned due to ethical and safety concerns. Therapeutic cloning, however, holds immense medical promise. Here, a patient's cell nucleus is placed in an egg, forming an embryo that's never implanted. Its purpose, to harvest embryonic stem cells, which can become any cell type. These cells, genetically matched to the patient, could revolutionize transplant medicine. Imagine growing new heart tissue, insulin-producing cells, or nerve cells for healing. Therapeutic cloning isn't about making people, it's about healing them. While it raises ethical questions, many see it as a powerful tool against disease. The line between healing and replicating is clear. One path creates life, the other restores it. The promise is hope, not duplication. If cloning is copying, genetic engineering is editing. CRISPR, a revolutionary tool, lets us cut and modify DNA with precision. It's like find and replace for our genetic code. CRISPR is transforming agriculture, creating hardier, more nutritious crops. In medicine, it offers hope for treating genetic diseases at their source. Therapies are already in clinical trials, editing cells to fight cancer and inherited disorders. This is personalized medicine, fixing the root cause, not just symptoms. The power to edit life's code is here. The question is how we use it. The future is arriving faster than we imagined. In 2018, gene-edited babies were born in China, proving the technology exists, though the ethics remain hotly debated. Therapeutic cloning has led to advances like induced pluripotent stem cells, which can become any tissue without creating embryos. Scientists have grown organoids, miniature organs, to study disease and test drugs. Lab-grown organs and tissues are moving from science fiction to reality. Gene therapies are curing blindness and halting deadly diseases. These breakthroughs offer hope and healing, not just treatment. We are rewriting the rules of medicine. The possibilities are extraordinary, and so are the responsibilities. With the power to copy and edit life comes a heavy responsibility. Cloning and genetic engineering challenge our ideas of identity and individuality. There's a risk of creating a genetic divide, where only the wealthy can afford enhancements. This could entrench privilege and create a biological caste system. The line between healing and enhancement is blurry. Who decides what's a defect and what's normal? Science fiction warns us of a future where genes determine destiny. We must ensure our pursuit of perfection doesn't erode our shared humanity. For the first time, we can steer our own evolution. The choices we make about cloning and genetic engineering will shape humanity for millennia. We hold the tools of creation, and the responsibility that comes with them. Will we use them to heal and uplift, or to divide and diminish? Science fiction offers cautionary tales, reminding us that progress must be matched by ethics. We need open global conversations about the future we want. The universe has given us consciousness, and now, the power to change ourselves. Our task is to use this power wisely, with humility and care. The future of humanity is not written in the stars, but in our genes. And we are the authors.